Okay, so today I will tell you a little bit about M13 phage, which is a flexible rod and can move around in the fluid and, of course, infect E. coli. So actually it's a famous disease, a sexual disease, because this phage only affects or infects male E. coli cells. Actually, it absorbs onto the tip of the F. pilus, and after absorption, the F. pilus is retracted, and the phage is disassembled. The DNA enters, the single-stranded DNA enters the cytoplasm. Then replication and protein synthesis is started, and first progeny DNA is covered up with a cytoplasmic DNA binding protein, which is replaced then by the coat proteins. There are 5 different coat proteins. All these 5 coat proteins insert into the inner membrane of E. coli, and we will focus, of course, for this interesting process, how these proteins actually insert into the membrane and then bind to the single-stranded DNA to extrude or secrete the phage. There is an additional protein in the outer membrane, also encoded by the virus, and this forms a huge pore which lets through the phage particles through the outer membrane. So it's basically a secreted phage. There is no lysis of the cell involved. Okay, the process of infection goes very fast, and here I show you some pictures of the process done with an atomic force microscope in our lab. So if you have uninfected cells, you see no phage, but shortly after infection, like 5 minutes already, so some phage come out at the tip of the cells. At 10 minutes, you see there is really the phage come out all over the cells, even at the lateral side. And at 60 minutes, there is, let's say, the boom of production of phage, and there is a lot of phage produced per cell, up to 1,000 particles. So it's a very efficient and fast process. Okay, so we are now concentrating on this actual event, how these coat proteins come into the membrane. And early on we found that this event is accompanied by a host factor, a host protein, which is in the inner membrane of E. coli. And this name of this protein is still derived from the sequencing project of unknown function genes. So the name is YTC. The YTC protein is an inner membrane protein which has a large periplasmic domain, and then it has in total 6 transmembrane regions, and it interacts with a coat protein and the major coat protein, for instance, and I will focus on this, how actually the major coat protein recognizes and binds to this YTC protein, and then it's released after translocation. So this N-terminal domain is actually translocated across with the help of YTC, and then it's released into the bilayer. So you have 3 events in steps going on in this process. So we could actually isolate the YTC protein and reconstitute into proteoliposomes and study the process in a pure form. So the in the membrane, or the proteoliposomes, which are then formed, only consist of poor lipid and the purified YTC protein. Then we purify the coat protein, and under conditions where the coat protein is soluble, we can add the coat protein to the outside of the proteoliposomes and follow the translocation and insertion of this protein into the membrane. So this actual nice process, so this is the structure of the YTC actually. The periplasmic domain has been crystallized, and so it's a structure of a so-called beta sandwich structure. And of course, the transmembrane part is still on due to crystallize. Hopefully it does soon. Okay, so this is basically the assay we use. So we purify the PF3 coat protein, and we have the proteoliposomes with the YTC protein reconstituted. Fortunately, the orientation of the YTC protein, as we checked it, is more than 90% in this orientation. So actually the outside represents the cytoplasmic phase, and the periplasmic phase is inside the proteoliposome. So this is actually, we can actually mimic the insertion process quite well. So now we labeled with fluorescent dyes the coat protein, for instance, the N-terminus of the coat protein, and we label also the YTC protein with another dye. So if the two dyes come together, we can measure FRET. So this was done by a graduate student, Sophie Winterfeld, in the lab. Actually, she could observe the FRET events during membrane translocation. So placing, of course, the fluorescent dyes in the adequate positions. How is this done? This is, of course, the drawing of a single molecule, but we also attempted to observe actually single events, so in single molecules. So we move into a system where we have a confocal microscope, where we can have a confocal volume which is observed by the fluorescent detectors. So as the proteoliposome swims through the confocal volume, we observe the events which happen actually on this proteoliposome. So we detect the fluorescence of the acceptor, of the donor, and eventually also the FRET signal. So this is detected from the beam which you shine on the confocal volume as these particles diffuse through. So we have about 50 milliseconds to observe. This is about the time a proteoliposome requires to diffuse through this confocal volume. So in this time span, we can look whether the coat protein actually approaches YTC and comes close, the two dyes come close to each other. This would mean that they actually are translocated, and if we follow the path, we can even see how fast the protein crosses the membrane B layer. Okay, so these are the results where we observe the distances. So the distance is listed down here. So you have, here is the distance. So you have here 4 nanometers and about 7 nanometers. And we now have 2 different cases. We have either the case where we see translocation. So this is the case I just showed you, where the one dye is on the N-terminus of the PF3 coat protein, and the other dye is on the periplasmic domain of YTC. And these cases show you that you have a very short distance, so the two come together, and you see you get a high energy FRET signal. If you make the control when the dye on the coat protein is at the C-terminus, which never translocates, you never get high FRET signal, you only get low FRET signal. So this is about the distance of 7 or 6 nanometers. And these 6 nanometers match about the distance the membrane has. So all these dyes can never reach the two. Now we put the periplasmic dyes on YC on different positions. So after the first transmembrane, third transmembrane, or fifth transmembrane, they all behave basically the same. So of course, these short distances you cannot really distinguish very nicely. They probably form a bundle because all the transmembranes form one structure. And then, of course, the PF3 dye, bound dye, come very close to either of these transmembrane regions. So, of course, you can make another control and place just YC dye on the C terminus. So then, of course, these guys which translocate separate each other again. And only these come close which do not translocate. So the C terminal dye on PF3 then comes close, the red ones, and that's what you actually see. That's the high energy thread now is just the collision on the cis side of the proteoliposomes. And these ones which are translocating are quite interesting because they show both. And this is consistent because if you add the protein, first the N-terminus comes close to this label on the cis side, but then it gets translocates and is moved away. So you have very few high energy signals and usually more of the low energy signals. Okay, now this is a statistics of many of these events as they swim through the confocal volume. But we can also look at individual processes on one. How does this actually look? And these are then the traces. This is a summary actually of the events. When actually here you see the distance, so these are all the far. There is always the membrane in between, and here are all the signals when they are on the same side of the membrane. So either they translocate like these, or they just collide on the cis side. And you clearly see this is the membrane in between, so you can distinguish the two sides just by these thread signals. Okay, now the single traces we have. So you just follow in time, so in millisecond what happens, and you can show here in the distance, if you draw the distance, you can show that it comes from about 7 nanometers down to 3 nanometers or 2 nanometers close, and then it separates again. So that this exactly matches the process we initially proposed, that the two dyes come together, YC and coat protein, and then they go apart again. So they are released. And here you see the release structure. So, and these are the different positions in the periplasmic regions. For all of these positions, we are about get the same kinetics, that the two dyes come together in about 10, I would say 10 milliseconds is the binding, and then the moving away is a little bit slower, I would say. Here's a control where the two cannot come together, and of course this just stays on one side, also on a single trace. Okay, so that shows that the speed actually is quite fast, and you can calculate this insertion time, so the speed from the 6 nanometers to the 3 nanometers distance, which is about then, about 5 to 10 milliseconds. If you vary now the temperature, this is done all at 20 degrees, so if you vary the temperature and do the process at 15 degrees, of course you cannot really do it at 0 degrees because the operators will freeze to death, and also the electronics do not really like too hot temperatures, so we did a small span of, let's say, livable temperature differences, but you see clearly the effect of insertion that it's temperature dependent, whereas the collision is practically not temperature dependent in this range, so the difference is too small to really detect. But this is quite clear that it's kind of an enzyme which has to, actually the YIT-C has to move. We have done also fluorescence studies where in the YIT-C protein where we can see that it moves inside YIT-C during this process. So we can actually characterize these different states and the process itself just by Fred. Okay, so that's of course a pure in vitro system, so we ask us the question whether we can also show that PF3 and YIT-C protein come close together in vivo. And there are of course some methods where you can follow in vivo contacts, and a very nice method is with disulfide. So you actually engineer at different positions cysteines in the PF3 coat protein, so only one cysteine in the PF3 coat protein, and then at different positions in the YIT-C single cysteine mutant. So we went actually in the practical course, we made lots of mutants, so every student had to make one mutant. We collected more than 100 single cysteine mutants. So we went through all these transmembrane domains, each position, and then asked the question, who contacts whom? So if we have cysteines at the middle of the transmembrane, we see contacts, two contacts here in the middle of TM1, TM3, TM4 one position, TM5 two positions, and usually they actually are in the distance of one helical turn. So of course now we can go to the other positions here and here, put here a cysteine and look for other contacts, and we see a similar thing. This is how these data are generated. So we have the coat protein expressed for a very short time because this event is very fast, happens only during the first, let's say, 30 seconds after synthesis. Then the protein is in the membrane, sees YIT-C, and is released again. So if we want to see a crosslink, a functional crosslink, we have to work very fast. So they are pulse labeled for 1 minute, and then you look what happened in the cell. So this is live E. coli, pulse labeled, 30 seconds. You open the cells, and you look for the membranes, whether you see higher molecular weight bands after immune precipitation. So you precipitate against the coat protein. Of course, some is dimeric because you have a cysteine in the neighbor, but you see higher contacts, higher crosslinks. And these crosslinks are in the size of YIT-C, which spans here, and are also you can immune precipitate the same bands with a YIT-C antibody. And actually you see here a small doublet. Here is only the upper band. So this is the band which is YIT-C and bound PF3 coat protein, which is just linked by a disulfide. So that's how these, we went through all these combinations of the coat protein. And so we went through all these contacts, and then we just looked the upper band. You see the upper band appears at position 14, 17, as I showed you before. So that's how we collect the data. And we went through all these transmembranes. And if we have now the cysteine at the blue position, we get two contacts in the periplasmic leaflet. If we have the cysteine at the green position, we get some in the cytoplasmic leaflet. This one is detected by both, and this one is detected by the red and the blue cysteine of the PF3 coat protein. So you can actually match, look, what is the interacting surface of the YIT-C during this membrane insertion event. Okay, that's what I wanted to tell you. And these are the people graduate student who made the AFM studies, Martin Ploss, and also the FRED studies is done by Sophie Börsch and Annika Schönbauer. Thank you very much.
Okay, we have time for one or two questions.
When you make a cysteine cross-link experiment, you are just locking something that should block transfer, right?
Yep.
Because the
You
cysteine
lock
is a cross-link.
yes.
Did you? So you should in fact accumulate all these things in the membrane.
No, because you are just labeling for 1 minute. You only have these contacts which you have during 1 minute. After this minute, it's TCA precipitated. Nothing happens anymore. And you can make the proper controls, of course, with out cysteines and
In the E. coli cell, is the YID localized randomly over the cytoplasm over the membrane or in specific locations?
You mean if it's distributed around?
Sorry?
You mean whether it's distributed all around the cell or whether it's at the poles? No, it's distributed all around.
Randomly.
Randomly, so if you label with GFP what we have done, you see you see a nice signal seam of the whole cell. So I wonder, why, for instance, the phage come first at the poles and then later at the lateral side, but I have no good answer for that. Maybe you have a good idea, but of course the process of phage extrusion is different than just getting these coat proteins across the membrane.
Okay, thank you very much. And now we move on to